we discuss about the quantitative real-time PCR. The quantitative real-time PCR is a widely used technique in biotechnology where the polymerase chain reaction or amplification products are analyzed at each cycle. So they are analyzed as the procedure progresses, that is in real time, as well as the template is assessed or quantified at each cycle, causing the quantitative real PCR to be a very strong technique for use in biotechnology. One of the application is to assess the gene expression, where the total RNA or messenger RNA is taken as the template, converted into cDNA, and then cDNA act as a template for the PCR reaction. So we can actually quantify the amount of RNA which are present in different cells, probably the cells isolated from different locations, the cells in a diseased or a non-diseased state, or cells which are at different concentrations. So the amount of RNA can be assessed. Other applications, qPCR applications, involve such as the genetic variation analysis. Now, genetic variation refers to differences in the DNA sequence encoded within our genomes. So, these differences we know over the evolution, there is accumulation of the various kind of variations in the genomes, and therefore the genome of individuals, they vary because of these variation that is there. So we can assess these variations by qPCR as well. The genotyping technology detects the genetic variations such as the single nucleotide polymorphisms or SNPs, the copy number variants, the insertion or deletions or called as indels, as well as the somatic mutations that can lead to major changes in the phenotype. So the genotyping technology, it can detect these kind of variations, and these variations can be quantified. Again, you know, the different DNA or the RNA, they can basically be quantified and these things can be assessed. For example, we take the mutation detection. So what happens in mutation detection is that it is showing the usage of a Tachman probe mechanism that we had discussed. So in the somatic mutations, some 0.1% of mutations can occur. However, the 99.9% are the wild type genes. So how to detect that 0.1% where the mutation has occurred? So what we do is we design these probes which are the allele specific probes. So these allele specific probes are going to bind to only the mutated gene which is there. So they are very specific. Again, they carry the reporter as well as the quencher, and then you have got the primers which is there. So this Tachman probe is basically locus specific. The primer is what is the again the allele specific primer that can be there. So this is the gene specific probe. It is going to bind to the mutant gene as well as to the real time gene. However, this is not going to, the primer is going to bind to only the allele which is basically mutated. This is not going to bind to the wild type gene. In fact, in the wild type gene, we also use a blocker primer. So this blocker is going to block this area so that even non-specific binding does not occur. So we use allele specific primers and locus specific probes. So these probes are going to bind to both the mutated gene as well as the wild type gene. Allele specific primer is only going to bind to the mutant gene, and then there is a blocker also which is binding to the same place where the allele specific primer binds, so that even the non-specific binding cannot occur here. Now when we carry out the PCR, then the polymerase is going to synthesize and extend this primer. It is going to remove this probe because of the 5' 3' exonuclease activity, and therefore it is going to give the fluorescence. However, because the allele specific primer is not bound here and this is a blocker which is bound here, no synthesis occur here, and therefore there is no fluorescence that can be detected here. So when the synthesis is occurring, the fluorescence is released here that can be detected. So there is no background, only the mutated gene can be, and therefore you can actually quantify the amount of mutations, and you can actually detect the mutation also. Because if there is no somatic mutation that is present, then there will be no fluorescence that would be detected. But if there is a somatic mutation that is present for the one which you have designed the allele specific primer, you can detect the fluorescence, and the detection of the fluorescence will tell you that the mutant allele is also present. So again, this is a very important and application. The somatic mutant detections, which are present in very, very less amount in DNA, but still this procedure is so sensitive that it can detect these mutations by in the cells which are there. Another application, or basically an analysis method also, is what is termed as the high resolution melting curve analysis. So apart from the Tachman probe, these high resolution melting curve analysis can be also done just like the for the analysis of the cyber green fluorescence detection. So what happens here is that approximately 80 to 250 base pair fragments of genes are amplified using PCR in mixtures that contain a high performance double stranded DNA binding dye. So just like cyber green, but it is not a high performance, it is a normal dye. If we use certain high performance double stranded DNA dyes, then these dyes are going to bind to the double stranded DNA. And as we discussed for the cyber green dye, the amount of fluorescence that is bound is going to produce the fluorescence, and that can be detected basically. But what happens here is that we also combine this with a method of melting. So you know what DNA melting is? When the double stranded becomes single stranded, the hydrogen bonds are broken, and this happens when the temperature is raised. So after 40 cycles, the amplification products are annealed and are highly fluorescent. So the end product, it will be highly fluorescent because there the amplification products are annealed and the fluorescence is bound to it, that can be detected. Now real-time PCR instrument slowly ramps the temperature higher while simultaneously recording the fluorescence data from the amplicons. So what will happen? The double standard PCR product is having a very high fluorescence. Now as you increase the temperature, the double-stranded DNA molecules will start separating and become single-stranded molecules, releasing the fluorescence. Now, when the fluorescent molecules, the dye, when the dye is released, when it is not bound to the double-stranded molecules, it does not produce any fluorescence. Therefore, there will be a dip in the fluorescence that will occur. As the PCR products begin to denature, their fluorescence will slowly decrease as the fluorescent dye is released until the temperature approaches the PCR product's Tm. So the melting temperature, for a typical double-stranded molecule, there is a specific Tm. There is a point in the temperature point at which the double-stranded DNA starts separating. That is termed as the melting temperature. So this melting temperature is unique for a double-stranded DNA molecule. So as we see the dip in the fluorescence dye, we will see that just before the Tm there will be a very large dip in there. So very close to Tm, a dramatic decrease in fluorescence is observed as the sample transitions from the double-stranded to a single-stranded DNA. So you can see this is the melting curve basically. So gradually the fluorescence is decreasing. As it reaches near to the melting temperature, the double-stranded DNA will now become single-stranded DNA. So there will be a sharp dip in the fluorescence, and then there will be, in single-stranded, there will be no fluorescence at all. Only the background fluorescence, but there will be no template fluorescence at all. So this is termed as the melting curve, and when we use high resolution dyes, this is termed as the high resolution melting curves. So when you compare the melting curves of two double-stranded DNA molecules, then depending upon the Tm, because the Tm of a mutated gene will differ from the Tm of a wild type gene, therefore you can again detect the mutations or SNPs or indels, and you can use this method to detect the genetic variants as well. So this is again a very, very powerful technique where the quantitative PCR mechanism is clubbed with the DNA melting mechanism, and both the mechanisms are used to detect the differences in the double-stranded DNA molecules. And this method is so sensitive that it can even detect, there will be a difference in the dip point, whatever this point is, there will be a difference even if there is a single nucleotide difference between the two double-stranded molecules. Their melting curves would differ. So the differing in the melting curve is going to help us to detect the mutations or SNPs or indels or whatever the genetic variants are there. So this is one of the application again for the quantitative PCR, and I gave you an example how it can be utilized. Other application includes, for example, the analysis of small microRNAs or the non-coding RNAs. So microRNAs are small non-coding RNA molecules that direct the post-transcriptional suppression of gene expression. You can again refer to the molecular biology sessions that we had conducted, where we discussed in detail about the microRNAs, the small interfering RNAs, etc., and how they cause this gene silencing, the post-transcriptional suppression of gene expression. So you can go through that. It discussed how these microRNAs are formed and what are their application as well. Now, if we want to analyze these microRNAs or non-coding RNAs in a cell, the quantitative PCR mechanisms again comes very handy. The various methods how it can be used is in the microRNA profiling. For example, identify differentially expressed mature microRNAs using quantitative PCR, and it is used in applications like oncology, metabolic diseases or autoimmune diseases. Under these diseased states, certain microRNAs would increase in number or certain microRNAs would decrease in number. So the microRNA profiling, just like we explained for how for it used for the RNA, so we reverse transcribe the microRNAs and then detect their presence or change in the number of expression, and then we can basically do the miRNA profiling in these diseased state. The microRNA can be quantitated. The specific quantification of mature microRNAs using quantitative PCR can be done. So if we are not comparing two states, we can quantify the mature. If we just not detecting, we can quantify it also. MicroRNA validation can also be there. We can confirm the microRNA profiling results using quantitative PCR, that whether truly that microRNA was present or its number was altered. We can do that by quantitative PCR. We can also do the microRNA functional analysis, because microRNA mimics and microRNA inhibitors for functional studies can be used. So if we use microRNA mimic, we can actually see whether there is a what change do we observe in the number as well as in the detection of the different microRNAs in the different conditioned cells, and therefore we can use it for the functional studies as well. Similarly, for the non-coding RNA analysis, the reliable detection and quantification of non-coding human, mouse or rat transcripts longer than 200 nucleotides can be detected. So basically, the small RNA analysis, detect and measure any small RNA, the pri-RNA analysis, which detect, measure the primary microRNAs. So if you go through the molecular biology lectures, you will see during the synthesis of microRNAs, there are primary microRNAs that are formed. So one can detect and measure these primary microRNAs as well from different species such as human, mouse and rat species. So in the quantitative PCR applications, some of the applications I have discussed. However, there are a number of applications where this quantitative PCR or real-time PCR can be employed. Majorly, it is employed in the gene expression analysis. Then it is also employed for the genetic variant analysis, where even the Tachman probes or the high-resolution melting curve methods can be used to detect the differential presence of these genetic variations. And as well as for microRNA, non-coding RNAs can also be detected. There is also what we do is called as the multiplex real-time PCR. So what happens in multiplex real-time PCR is, because in this case we are using different fluorescence chemistries, so we get those molecules which can produce different fluorescences, and therefore in one tube, in one reaction, we can actually detect different RNA molecules that are expressed. For example, if we have taken total RNA, then total RNA we are using real-time PCR, we can use the primers or where the double-stranded different dyes can be used, or we can use the different Tachman probes which are bound to different fluorescences, and therefore they are locus specific for two different genes, two different fluorescent reporter molecules can be utilized, and in one reaction mix two different expressions can also be studied. So this is termed as the multiplexing. So multiplexing is a technique in which more than one target is analyzed in the same real-time PCR reaction. Each target is distinguished by a particular dye conjugated to the fluorogenic probe or primer pair specific for that target. So this multiplexing gives us an advantage because typically these reactions are used to amplify a normalizer gene and a gene of interest in the same reaction. So when we talk about the relative method of quantifying a target DNA, we said that we use co-amplify certain steady state genes. So if we want to use that steady state genes, we can probably use two different fluorescences, one for the steady state gene expression and one for our own interest gene expression, and simultaneously we can measure and quantitate the amount of both the things, and relatively we can calculate the concentration of the unknown target DNA. So multiplexing again gives advantages because there is less variability, there is more consistency in the results that we obtain. Multiplexing a normalizer and gene of interest in the same tube eliminates the well-to-well variability that can arise if those same two targets were amplified in different, even if it is adjacent but they are in the different gels, different wells. The less reagent usage is there is less cost. Multiplexing requires fewer reactions to analyze the same number of targets. There is a higher throughput for this multiplex PCR. The more targets can be analyzed per real-time PCR run and per sample. Obviously, this will reduce the cost as well because less amount of reagent usage is there. So, there are many advantages that multiplex PCR gives in. However, still it depends upon the purpose that you utilize, because again there are some disadvantages which are associated with it because you are using two different dyes. They can interfere with each other, the quencher molecules also that you are using. So you have to be very particular about what dyes you are using, what probes you are using, and it has to be designed very and planned very properly so that this becomes an advantage to us. But there are many advantages of the multiplex PCR that I have discussed. So that is all about the quantitative PCR or real-time PCR, the procedure, the ingredients that we use for it, how do we analyze the data, what is the application of quantitative and real-time PCR, and what are some of its modifications like the multiplex PCR that we use has been discussed in this session. Now what happens is that apart from the polymerase chain reaction, so when we started with polymerase chain reaction, the idea was basically to prepare the number of copies of a particular gene. So whatever gene or region we are studying, we can detect its present, because if we get the product, that means that is present. If we do not get the amplified product, that means that is not present, whatever primer specific you are using. So we can detect the presence or absence of a gene or a gene of interest or the RNA if we do the reverse transcriptase. We can also increase the copy number, because sometimes, like I said, in recombinant DNA technology, we were doing molecular cloning. So this is a very quick method. We utilize this method. We employ this technique for creating copies of the region of interest really quickly, and a large number of copies are produced. And then we can work on the the functional aspects, or what we translate it into the RNA and study functional studies, etc. what can be done. But still, sometimes we need to determine the exact sequence or order of the nucleotides or bases in a DNA molecule. We cannot do away with that. We can have the product, we can put it for functional analysis, we can clone it, we can study it, but even if we clone it, then one of the method why we want to clone is to sequence it. Because the entire information that is contained in the DNA is in the order of the nucleotides that exist. What is the pattern of A, T, G, C, the 4 base pairs? How are they arranged? The entire information is contained in this sequence. So, if we want to analyze it for further studies, we need to determine the exact sequence, and the technique that is used is termed as the DNA sequencing. There has been exponential improvements in sequencing DNA techniques more rapidly and more cost effectively. The entire genome can be sequenced for about $1,000 in less than a day. And now it has been further improved, and we anticipate that probably in a matter of few hours, the DNA, our genome, whole genome I am talking about. So we take one of our cells and we just sequence the entire genome in a matter of some hours. In a near future this will happen for as less cost as $100. So, obviously there is some constraints where we talk about the ethics in this DNA sequencing, but DNA sequencing has the methods has improved, it has become more rapid and more cost effective. In a relative sense, I tell you that the Human Genome Project, it costed somewhere around $13 billion, and it took somewhere around 10 years to sequence the human genome when it started with. So when it started with, it was a 10-year human genome project and it costed around $13 billion. And now the genome can be sequenced in a matter of few hours for $100. So you can imagine how the sequencing techniques and technology has evolved, and it is so important for the biotechnology which is there. Now, one of the method that I discussed today is the Sanger sequencing method. It is an old method, but it is still very, very widely used. The entire technique has been automated, but still, the basic principle we need to understand, which is basically the sequencing by synthesis. So, as the complementary strand of the DNA template that you want to sequence is synthesized, it is basically read. So the order is read. So what happens here is, this is also termed as the chain termination method. So what is basically there is that when we are synthesizing, so synthesizing DNA we have already talked about. A typical mechanism which is harnessing, because biotechnology is the technology derived from the biology. So the biology is DNA replication. Technology is PCR. We have talked about PCR. This is basically synthesis of DNA. So we have a single-stranded DNA, we want to synthesize its complementary DNA. So what do we add? We add DNTPs. So DNTPs will be added and the polymerase is going to synthesize the complementary DNA. So what do we do in the chain termination method is that we add the DNTPs, but one of the DNTP we add as the ddNTP. So it is a dideoxynucleotide phosphate. So deoxynucleotide phosphate is the normal ones, and what is the difference between the DNTPs and ddNTP? That at this position where the hydroxyl group is there, it is replaced by a hydrogen group. So this, when the hydrogen group is added, it becomes a dideoxynucleotide, ddNTP, nucleotide phosphate, and this ddNTP terminates the chain synthesis. So no more synthesis, the polymerase is unable to attach because it does not have the 3' hydroxyl group. So because it does not have 3' hydroxyl group, it is unable to synthesize and therefore the chain terminates. So wherever this ddNTP is added, the chain will terminate. In a particular reaction, because you have normal DNTPs and you have got altered this ddNTP as well, you will have a series of fragments where the chain is terminated. So if you run this PCR amplification up to a particular number of cycles, you will probably have a 1 nucleotide resolution, that you will have number of fragments which are terminated at 1 base pairs apart. And when you run these samples on the gel, so what you do is you take 4 reactions. In one you add DDATP, in another you add DDGTP, DDCTP and DDTTP. So whatever happens, there will be a series of fragments here which would have been terminated at every A that has occurred in the sequence of the DNA. Every G, every C, every T. And as I said, when you run it for a number of cycles, you will have the collection of fragments which have stopped at DDATP and with the 1 base pair resolution. So you will have all the fragments, length, sizes. So when you run simultaneously on a gel, in a very vertical gel, long gel, high resolution gel, there will be 1 base pair resolution. You can actually read the sequence, because the A has been stopped, then G, then C, then T, then T, then A, then G, C. You can just go from down to up and you can read the sequence. So this is what the Sanger sequencing and the chain termination method is. Now this is principally how it happens. We no longer do it in the 4 tubes. We do it in a single tube. We attach the DDATPs with different fluorescences, and we do not do a normal gel electrophoresis, we do a capillary electrophoresis. So the entire system is now automated. So the entire system is automated. In one tube, all the reactions are happening. Now they will arrange, when you run in the capillary, they will arrange according to 1 base pair differences, and these different dyes will be detected by the laser. So when the laser comes, the fluorescence will occur and there will be a detector, and it will give you this kind of a chromatogram. And just by looking at the chromatogram, you can read the sequence. So this is the Sanger sequencing method. This is very important method. It is still being used, although it is an old method. It was discovered in 1977, and this was such a revolutionary method, technique, that he got Nobel Prize in chemistry. Sanger, Frederick Sanger, he got the Nobel Prize. So this is a very revolutionary technique that is used. In the next session, we'll talk about the next generation sequencing method platforms, the NGS platforms, which are nowadays used, which give a single, where even from a single molecule one can get the sequence, and how it has become so rapid and so less. This has got a limitation that only 600 to 700 base pairs can only be sequenced. Now our whole genome are in MBs. So, if we are sequencing 600, 700 base pairs in one reaction, obviously it will take a lot of time. It will cost a lot of, chemicals, etc. So, to cut down the cost, etc., now newer platforms have been rising. There are NGS platforms that we discuss in our next session. So, that would be all for today. Thank you.